Okay, so welcome to the last chapter for the semester. So this is chapter 19, where we're going to focus on genetics and inheritance. With this chapter, we're going to go through kind of a basic introductory section, I guess you could say, where we're going to talk about genes, we're going to talk about genetics, we're going to talk about chromosomes, chromatids, and basically go through like an introduction. So talk about alleles, etc. And then after that, we're going to focus on patterns of inheritance. So how we actually inherit our genes from our parents and kind of what those patterns are and the different probabilities that exist with whether or not you get a gene from a certain parent and how often that could show up, say, in your offspring. And so for the first video in this chapter, we're just going to kind of go through some introductory terminology and a basic introduction for genetics. And so this is just a really quick and dirty overview of genetics. And so in our cells, we've already talked a lot about this. We have genes. Right? And when we talked about mitosis and meiosis, we've mentioned a lot of these terms already. And so genes are on your chromosomes, and there are basically DNA sequences that are or that have the instructions for building our proteins, right? So we talked about a gene being transcribed and translated from DNA to messenger RNA to a protein. Now, genetics is the study of genes and their transmission from one generation to the next, so their inheritance patterns. And now, if we look at all of the genes, all of the DNA that is in an organism, that's known as its genome. So, whenever somebody says the genome has been sequenced for a particular organism, we're basically saying that all of the DNA has been sequenced and we know what that structure looks like in terms of its base composition, base pair composition. Now, your DNA is made up and consists of chromosomes. So your DNA is found on your chromosomes. And the DNA, or I should say the number of chromosomes, is dependent upon the species. So for example, humans have 23 pairs of chromosomes. So we have a total of 46 chromosomes. 23 of those pairs come from your father. The other 23, or I should say not pairs, but 23 chromosomes come from your father and 23 chromosomes come from your mother. And on those chromosomes, you have your genes. And so a chromosome can be made up of, or it does consist of, several genes. With the 23 pairs of chromosomes found in humans, 22 of those pairs are known as autosomes, and one pair is known as sex chromosomes. And these sex chromosomes actually determine the gender, so whether or not you have an XY or you have a double X. So this would be your males and the double X would be your females. Now, for the autosomes and the sex chromosomes, each pair, so for each pair, you have one from your mother and one from your father. And we can actually, scientists can actually go in, or geneticists, I should say, can go in and perform what's called a karyotype. So they can take a cell that is about to divide, where the chromosomes or where the DNA has condensed into chromosomes, and they can stop that cell from dividing. They can stain those chromosomes to show the different genes, and then they can match those up, match the pairs of chromosomes up, and provide a visual display of the chromosomes for an individual. And this would show all the pairs of chromosomes lined up side by side. And so this outcome is known as a karyotype. And so here's a figure of a karyotype. So you have chromosome 1 all the way to chromosome 22. So those are your autosomes. And then your sex chromosomes here are pair 23. And so this individual is a male. It has an X chromosome and it has a Y chromosome. Now notice all of the banding patterns. And notice that with each pair, they are relatively the same size and shape, and they have the same banding pattern. So like this one, right? You have that red, then you have green, then red, then green, and red, right? And then here you have like yellow, red, yellow, green, and the same thing here, right? Those typical patterns. Now each band represents a gene, a segment of DNA that's going to code for a particular protein. Okay, now these pairs, remember when we talked about meiosis, are known as homologous chromosomes. So they look alike, same size, same shape, same banding pattern. One member of these homologous chromosomes comes from each parent, right? So one member of the pair. So this, let's say, could be the father. This could be the mother, the chromosome contributed by the father and the mother. Okay, because excuse me, because each chromosome is contributed by a different person, a different parent, just because it's the same gene does not mean it's the same version of that gene. And so the different versions of the gene are known as alleles. So for example, the father could have passed on brown eyes version of the gene. The mother could have passed on blue eyes, right? And so the combination of those alleles are going to determine what the offspring looks like. And so again, homologous chromosomes, are those pair that is inherited from each parent. They look alike, but they're not identical because they have different versions of the genes on those chromosomes. And the different versions are known as alleles. And then alleles arise from mutation. So mutation is the key form of variation in the natural world. Mutations will give rise to all of the variation that we see. Now, if an individual has two alleles that are the same, they are known as homozygous. If they have two alleles that are different, they are heterozygous for that gene. Okay, so for example, let's say you have, let's go back to this karyotype. Let's say we're looking at chromosome 13, so right here. And let's say we're looking at this bottom gene. Right, so let's say it has hair color. And let's say if you have the big B allele, you have brown hair. If you have the lowercase b, it represents blonde. Okay, so let's say this one comes from the father and this one comes from the mother. The mother passed on the blonde allele, the father passed on the brown allele. So these two alleles are different. So this is the heterozygous. Now, let's say if they were both the brown allele. So the father passed on the brown allele, the mother passed on the brown allele. Then they have the same allele, and so that's homozygous. Okay, so the same as homozygous, different alleles are heterozygous. Okay, and then the combination of those alleles determine the outlook or the outcome of what that character is going to look like. So the genotype, which is the homozygous or the heterozygous, determines the phenotype, so how you look. Okay, and we'll talk more about that in a couple slides. So for example, here we have a pair of chromosomes, so autosomes. Right, so here's your pair. And here we have three genes. So we have 1, 2, 3. Now the top gene is both purple, so they're homozygous. The middle gene is both yellow, so it's homozygous. The bottom gene is two different colors, two different versions of those alleles. So that is heterozygous. Okay. Now, where those genes are located is called the locus. And you have multiple places for genes. That's the loci. So the locus is singular and loci are plural. Okay. So you can say this gene is located at this locus on the chromosome. Okay. And then again, the alleles are the different versions of the same gene. And now, like we mentioned, the genotype is basically your set of alleles. So for example, you could be 2 alleles for brown hair, or you can have an allele for brown hair and an allele for blonde hair, or you can have 2 alleles for blonde hair. Okay, this would be your genotype. These are homozygous because they are the same, and this one is heterozygous because they are different. Okay. The genotype determines what your phenotype is. So for example, we're going to talk a lot about dominant and recessive alleles. So let's say this is a dominant, a recessive allele relationship. So the uppercase allele is generally representative of being dominant. Lowercase is usually recessive. So anytime you have a dominant allele, that is the character that is going to be expressed. So since brown hair is dominant, anytime you have that uppercase B or the brown allele, the individual will have brown hair. So because you have at least one brown allele, this individual will have brown hair, right? And it's homozygous dominant, okay? This has 2 alleles for the recessive allele. So there's no dominant allele, so it will show the recessive trait, which is blonde. And this is homozygous recessive. Okay, that's a REC for recessive. Now here in the heterozygous, you do have a dominant allele, and that's going to mask out the blonde. So this individual will have brown hair because the dominant allele dominates the recessive. It masks it out, so it has the brown characteristic. So of these genotypes, you have 2 phenotypes that are brown hair, or blonde hair. Okay, so the phenotype is the observable physical functional trait. So for example, hair color, which we've already talked about, or it could be like eye color, skin color, blood type, disease susceptibility, height. All your physical traits are generally underlain by the specific genotype that you have. Okay, so phenotype is determined by those inheritable alleles, but also environmental factors can affect those as well, and we'll mention that here in a little bit. Okay, so what we're going to look at now is how we can determine patterns of inheritance. So there's this analysis that was derived called the Punnett square analysis. And this allows us to predict patterns of inheritance. And so we can set up a Punnett square by following these steps, okay? So basically we have possible alleles of one parent placed on one axis and possible alleles of another parent placed on the other axis. And then we look at the possible combinations written in the grid. So let's say we have a square. We're going to divide it into 4 quadrants. So this is our Punnett square. Let's say we have a male that has brown hair and he is homozygous dominant for brown hair. So he has 2 uppercase or dominant alleles. let's say we have a female that has brown hair, but she is heterozygous. She has brown and blonde alleles, but because brown is dominant, she shows up as having brown hair. Now we want to know what is the probability that this couple has an offspring with brown or blonde hair. So whenever the genes are transferred to the sex gametes, so the sperm or the eggs, remember they're cut in half. So you're only getting one allele. So the male is either going to give a big B or a big B. The female can give a big B or a lowercase b because she has two versions. So we're going to write these up here. So we're going to put the father up here and then the mother. It doesn't matter. You can switch these. It all lead to the same outcome. And then you just combine. So you have a big B, dominant B, and then a dominant B. Same thing here. So dominant, dominant. You have a dominant, then you have a recessive. Then you have a dominant, and then you have a recessive. And so what this Punnett square tells us is that all of their offspring will have brown hair because all of them will have at least one dominant allele. However, 50% of them will have the blonde allele, so they'll be carriers for the blonde allele. And then in order for blonde to show up, you basically would need two heterozygous individuals or an individual that's blonde to mate with a heterozygous individual, right? So let's kind of show that. So you would have, right, your uppercase, 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 lowercase, uppercase, lowercase, and then your two lowercase. This would be your blonde individual. So in this scenario, 75% would be brown, right? So 1, 2, 3, and then 25% would be blonde, which is this one. So 1 out of 4. 25% will be homozygous dominant, which is this one here. 50%, so 2 out of 4, will be heterozygous, and then 25% will be homozygous recessive. Okay, so we can use the Punnett square to kind of get those averages. And so here's some other examples where this is the one we just showed here with the two homozygous, where you have the 25% dominant, 25% heterozygous, and then 25% homozygous recessive. When you have, say, a homozygous recessive with a homozygous dominant, all of the offspring will be homozygous and all of them will have the dominant phenotype. And so you can kind of look through these different scenarios for the Punnett square and figure out kind of what the probability of having an offspring with a specific trait could be if you have offspring with a person who has these alleles compared to your alleles. But now this Punnett square is for this situation is only working for, say, dominant recessive alleles. And we'll talk about some variations in the last video for this chapter where you will have variation from dominant recessive to where there's kind of these other patterns of inheritance. Okay, so that's it for this first video. The second video, we're going to go in and talk about the person who established these basic principles, who kind of developed the Punnett square, and look at the laws that he figured out. So that person is Gregor Mendel. And so he determined the law of segregation and the law of independent assortment, which we talked about during mitosis and meiosis chapter. We're going to talk about exactly what those are, what those mean, how he figured those out. We'll look at the Punnett squares a little more in more detail, and we'll look specifically at one trait crosses, so like we just did with the hair color, and then we'll look at two trait crosses, so like if you have hair color and eye color, kind of wondering if you'll have brown and blonde, or brown hair, blue eyes, or brown hair, green eyes, etc. What's the probability of having two traits show up together? And so as always, let me know if you have any questions, if you have any concerns, and we'll see you in the next video.